Some bases in tRNAs can be chemically modified into rare bases, and so you can start out with one fairly routine nucleotide, such as G, A, U, or C, in RNA, and then it can be chemically modified, for example, from a uracil to a ribethymidine or a pseudouridine. So these are structures that are chemically similar, but then are enzymatically modified to become different bases, what we call rare bases, which are indicated here in red. All right, so modification of tRNAs include, you can start out with introns in eukaryotes. Whoops. These introns can get spliced out. You also get the ends trimmed, and then the what do they call it? The amino acid attachment site, which is always CCA in the 5' to 3' direction, gets added on. So the CCA is not encoded into the DNA of the tRNA gene. It's added on later. And then finally, in a final step, you can get chemical modifications of these rare bases. Oh, I also wanted to say that in both eukaryotes and prokaryotes, tRNAs may be transcribed as a single unit, and then they are cleaved in process to yield separate tRNA molecules. And when a physically clustered group of genes is transcribed as a single unit, that's called polycistronic RNA. And this is fairly common in prokaryotes. It's much less common in eukaryotes, but it does exist. And one example is with the tRNAs. All right, so ribosomal RNAs. Ribosomes, again, are ribonucleoproteins. They are comprised of both protein and RNA, and the RNA component of the ribosome is called ribosomal RNA, or rRNA. Ribosomes in both bacteria and eukaryotes are similar, a little bit different in size. They both contain a large and a small subunit, and within both subunits, so here's the large, here's the small, within both of these subunits is RNA, rRNA. So in the large subunit in bacteria, there are 2 rRNA components, one in the small subunit. In the large subunit of eukaryotes, there are 3 rRNA components and 1 in the small subunit. Just a note, these subunits and the rRNA are they're denoted with an S that indicates their size, and the S stands for Svedberg units, which is a measure of sedimentation during centrifugation and is also correlated and indicative of molecular size. So moving on. All right, so in prokaryotes and eukaryotes, ribosomal RNAs are transcribed in the in the nucleus and are processed within the nucleolus, which is this dense material within the nucleus. And both prokaryotes and eukaryotes engage in polycistronic RNA transcription of rRNAs similar to the tRNAs, where you have multiple rRNA genes clustered together, transcribed as a single unit. They get methylated and then cleaved into intermediates, which are then further processed to yield mature RNAs. And a similar process also happens in eukaryotes. The processing of ribosomal RNA is accomplished by another RNA molecule called small nucleolar RNAs, or SNO RNAs. Nucleolar because they act in the nucleolus for rRNA processing, and they help assemble the ribosome. And so just to keep everything straight, we have small nucleolar RNAs that are assisting with processing and processing of ribosomal RNA, or rRNA, and assembly into ribosomes. And there's also small nuclear RNAs, and that's the RNA component of these spliceosomes, which are acting within the nucleus. So we're getting heavy into RNA land here. So now we're going to talk about a class of small RNAs that can act in what we call RNA interference or in cellular defense of prokaryotes, and that's referring to the CRISPR RNAs. So RNA interference was discovered by Andrew Fire and Craig Mello in 1998, and they received a Nobel Prize for it in 2006. And what they found, so we had previously known that antisense RNA, which is RNA that is complementary to mRNA, it's a messenger RNA, can bind to it and inhibit translation, and so that can be one way of silencing a gene. So we can talk about regulation of gene expression at the level of chromatin structure, such that when you bind your DNA so tightly with nucleosomes that that transcription is prevented. There's that level of gene silencing, but then you can also silence a gene at the level of the RNA, at the level of the mRNA, and occupy or destroy the mRNA to prevent it from being translated. And these processes are accomplished through small RNA molecules in a process called RNA interference, and that's what we're going to talk about. So it was previously known that antisense RNA can do this, just single-stranded antisense RNA, but Andrew Fire and Craig Mello discovered that when you use double-stranded RNA, it actually works even better. And so since then, the mechanism for this has been elucidated, and there's two types of small RNAs that are involved in this. One is small interfering RNAs, and the other is microRNAs, and they are similar but different in their function and their consequences. And so double-stranded RNA of the small interfering RNAs gets cleaved by an enzyme called dicer, and these small so these RNAs are, on the order of 20 to 30 nucleotides long. They're really short. So they get cleaved into these fragments. These fragments then become single-stranded and incorporate with a protein into a ribonucleoprotein called RISC, which is the the RNA-induced silencing complex. And this RISC component, this RISC ribonucleoprotein, recognizes, because this small interfering RNA has perfect complementarity with the messenger RNA that it's targeting. And that's because it was originally transcribed, it originated from the same gene that it is targeting. And so when the RISC interacts with this mRNA, it targets it for cleavage and degradation. On the other hand, there's microRNAs, and these are different genes from the genes they're targeting, and they include an inverted repeat that forms a stem loop structure after transcription in the RNA. This should be a pre with an E microRNA. Then it gets processed and cleaved. These arms get cleaved off, and then this loop gets also cleaved off, yielding just the stem. And this is done with dicer. As in the small interfering RNAs, a RISC ribonucleoprotein is formed from one strand of the microRNA. But here the base pairing with the target mRNA is not exactly perfect. And here it inhibits translation. It just sort of hangs on to the mRNA and just takes it out of commission, prevents it from leaving the nucleus, inhibits translation. All right, so that's RNA interference. We also have Pee-wee interacting RNAs that are specific to reproductive tissues, especially in testis and ovary, and particularly in the germline during spermatogenesis. These are also small RNAs, and they act to prevent transposable elements from jumping around in the genome and causing mayhem. So transposable elements are selfish genetic elements that can excise themselves from where they are in the DNA and move somewhere else. So they have copy and paste functions, and they have cut and paste functions where they can physically move themselves, or they can just copy themselves and then move their copies around. And depending on where this happens, this can disrupt gene function, and this is especially dangerous in germline cells. And so Pee-wee interacting RNAs inhibit mobility of transposable elements in germline cells. I just want to say that CRISPR is the next thing I'm talking about. CRISPR RNAs are basically double-stranded RNAs they're not double-stranded. They are in bacteria. They're derived from phage DNA, and it is a defense mechanism for bacteria against phage that might infect it. And so if a bacterial cell survives a phage infection, it will incorporate some of the phage DNA into its own genome in what we call spacers. And these spacers can then be transcribed and cleaved and processed. And they also interact with a protein and form a ribonucleoprotein complex that then targets phage DNA in the event of another infection and is able to cleave and destroy the phage DNA. And so CRISPR is actually being used as a mechanism for fighting Ebola in Ebola drugs. Oh, you know what? That's wrong. Sorry. It's not CRISPR. It's actually RNA interference that's being used. So these guys, these mechanisms, are being used in Ebola drugs, anti-Ebola drugs. All right, and finally, I want to talk about long non-coding RNAs. And these are RNAs that especially seem to be, I believe they're present in most eukaryotes that have been examined to varying degrees. And it turns out that we have large spaces between our genes and our genome. And in fact, only about 1% of the human genome encodes for proteins, but over 80% of it is transcribed. And often it's transcribed in these long, what we call long non-coding RNAs. They're long RNA molecules, and we don't always understand their function, but it turns out that they seem to be important in a large variety of ways, and one of which is actually the shutting down of the X chromosome to form bar bodies. So one example of this is a long non-coding RNA gene called XIS that's on the X chromosome, and when it's transcribed, it helps deactivate the X at specific time points during development.